The exercise for today involves two elements that we all use in our daily lives: water and air. We all use water and breathe air, so it's important for us to ensure that both of these staples of our daily lives are the best that it can be. If for some reason they are not a good quality, our health might be at risk. Today, for the quantitative examination of water, you will be using the information from your colony counts to determine the number of bacteria per mil that are in the 4 mil broth culture that I have prepared for you today. These are located in the center of your bench. To accomplish this, we will learn a very important concept that you will need to master in any lab, which is how to conduct a serial dilution. I've noted this on the board. By conducting a serial dilution and plating a known volume, one can estimate the number of bacteria the starting sample volume contains by simply counting the colonies that arise after spread plating and back calculating. Remember, a colony forms from one single bacterial cell, so by diluting out the initial culture and spread plating it, you're spreading out the individual bacterial cells across the surface of your plate. After incubation, these cells have replicated to the point that we are able to visualize them as colonies. Please refer to video 17 entitled How to Spread Plate an Organism. During this procedure, make sure to use a different tip for each of the transfers, as well as once a transfer has been made. You mix the container as well. In the case of the 99 ml dilution bottles, you will cap the bottle tight, shake the bottle, ensuring the organism has been distributed evenly throughout the solution in the bottle, and make the transfer into the next bottle or tube. In the case of the 9 ml water blanks, you will cap the tube securely, and using the vortex in the room, vortex the tube for a few seconds before making the next transfer. Each tube for the dilution has a dilution factor. Each dilution factor is multiplied by the next. Take note that the 0.1 mil you will be plating is another dilution you must factor in when you are calculating the dilution factor of the plate you will be inoculating. The next procedure we will discuss is the most probable number test. This test is used to examine a water sample for the presence of coliforms. You may have brought one in today. As you are all aware, coliforms found in water samples are a cause for concern at our public beaches and can often cause closures. They can also be problematic in our public water systems. I'm sure you have heard reports of mandatory boil water notices due to natural disasters that sometimes damage sewer lines, which then contaminate our drinking water. If you are ever in a situation as this and need to disinfect your water and you cannot boil the water, you can simply add 8 drops of household bleach to each gallon of water you have. The MPN procedure is conducted in the manner illustrated in your laboratory manual, Figure 7A. The MPN procedure is mainly conducted on small water samples and was the first procedure developed to test for coliforms. This procedure only estimates the presence of coliforms. This procedure is made up of 3 tests, these being the presumptive, confirmed, and completed tests. As this procedure requires many lactose tubes, some at double and some at single strength, and using various volumes of the water sample, we will be instead conducting a modified and scaled down version of the MPN test in lab. Be aware that to conduct the MPN properly, you will need to perform the test as it is illustrated in your manual. Since we will not be doing anything with the organisms we obtain from the confirmed test, we will not be conducting the completed portion of this procedure. The presumptive test is called this due to the fact that we are simply inoculating the water sample in a lactose tube and examining them for the presence or absence of acid gas production due to the lactose medium's fermentation. If a tube at this stage in the procedure has acid and gas after inoculation and incubation, we cannot conclude that this is solely due to the presence of E. coli, as E. aerogenes, which is commonly found in the soil, could also be causing this to be formed. Additionally, the acid and gas production could be due to two organisms working together to ferment the lactose. This is why we must conduct the confirm test in the next lab. The confirm test is accomplished by taking a tube of lactose that exhibits acid and gas and placing it on a plate that is divided into 3 sections of Eocene methylene blue agar. We will be placing in the 2 remaining sections prepared cultures of E. coli and E. aerogenes. After this has incubated for 24 hours, we will check on the result. If the growth from the water sample we tested produces a beautiful metallic green sheen, we can confirm that indeed our water contained E. coli. The completed test would then involve placing the organism on a new slant, as well as inoculating another lactose tube and getting acid gas once again. We have included the preparation cultures in the section of the plate so that you can compare your sample to a positive and negative control. The next technique we'll discuss is the membrane filter technique. The membrane filter technique was created to be an alternative to the MPN. Benefits to using this technique over the MPN is that this technique allows you to isolate colonies of coliforms and other microorganisms. It also allows you to then obtain those colonies for study, as well as allowing you to test a much larger sample of water. This technique will be demonstrated to you in the video number 17, membrane filter technique. This technique employs a vacuum to draw water through a membrane using a funnel system. It is the preferred method for testing large volumes of water. The medium we will use for this technique is MF endo broth, which is selective and differential for coliforms. The medium is selective as it contains sodium sulfite, basic fuchsin, and sodium deoxycholate, which inhibits the growth of gram-positive organisms. The medium is differential as coliforms produce a red color on this medium and non-coliforms are colorless. After filtration is accomplished, the membrane is placed on an absorbent surface that has been impregnated with this medium and incubated. The last test you'll be doing in this exercise is that of air quality. This part of the exercise is a take-home assignment. You will be given 1 plate of nutrient agar as well as a plate of sabouraud dextrose agar. The nutrient agar, as you know, is a general purpose agar, and the sabouraud dextrose agar is enriched for fungal growth. You can either place both plates in the same location or in different locations around your home. You will expose both plates to the air by removing the lid from the plate for a few seconds to up to 5 minutes. The length of exposure depends on how well the space is aerated. If you are holding it up to an air conditioning vent, you will only need to do so for a few seconds. But if you are placing it in your shower, since the bathroom usually has an exhaust vent, you may need to leave this plate open a bit longer. Return the lids to the plates after exposure, invert, and incubate at room temperature until you are able to return them to the lab. Upon returning to the lab, if there is growth, make note of what you see, and if there is no growth, you will incubate the plates until the next lab period. Remember, just because you cannot see growth after room temperature incubation doesn't mean there is no organism present. It just means that maybe the organisms will require a warmer temperature to grow. The incubator will simulate your body's temperature, allowing you to see what could potentially grow if left unchecked by your immune system.